Раз. Okay, let's start. So time is over, and in the next 3 hours we see the results, your results, and we really hope that this will be very interesting. And what will we do? And the first part involves 10 projects split between bioinformatics and biostatistics. Then we have a short 10 minutes break and another set of 10 bioinformatics projects. Each presenter team will have 5 minutes for the talk and follow it by a few minutes for the questions. And also we ask to join our jury team. And let's get introduced the jury members. First of all, it's Kostya Zaitsev. He works for JetBrains company. Then Edgar Galstyan from EPAM. Kostya Chernyshev from Boston Gin. Evgeny Bakin from IBRE team. And Nadezhda Pogodina from Genetech. And of course, our team joined to the jury too. And if you have any question about presentation, or you, okay. Following the project's presentation, there will be a 30 minutes break for us to select the winners, and then we come back in this hall and make some, I don't know how to say that, give you some prizes. Okay. Yuri? Yeah. First part is you.
Great. Yeah, thank you. And thanks to all of you for submitting your slides on time or almost on time, as always. And the first team to present their project will be the team that was working with predicting protein stability using deep learning. Please. Yeah, your 5 minutes.
Hello everyone. It's very pleasant to open these hackathon presentations. So our team is Wings team, as you already see. Our team names are there. Our team actually are also there. Our team leader are online. And today we'd like to present project protein structure prediction using deep learning. So why we do that? Because AlphaFold is there, and why we do that? We do that because AlphaFold has a good performance on the large proteins, but sometimes struggles predicting microproteins. And they are really important because many immune receptors are small proteins. So we live during COVID, so we know that immune system is very, very important. So we, sorry. And we try to use secondary structure in secondary structure we trust, because some small proteins don't have a tertiary structure, and also it's a good predictor for not always for protein function, but for protein-protein or protein-drug interaction prediction. The goals and tasks of our okay. Sorry. The goals and tasks of our work were to develop system, predict second protein structure using known open source instruments, and the tasks was to collect SOTA approaches, to compare them, to create pipeline, develop user-friendly service, and embed visualization function. So we use protein glue benchmarking. We have a really huge database with 11,000 of proteins, and they have benchmark for several tasks. We have enough time to complete 2 of them. It's prediction of secondary structure and predict solvent accessibility based on that, and three more. It's really good future plans, and we really hope that we have enough time to do that, perform that also. Yeah, so Elizaveta continues.
Okay, so yeah, as Zhenya has already said, we've tried different benchmarks, and we took the initial model which was made in protein glue, which was simply a BERT model, and it was our baseline. We managed to launch 2 different models which are called ESM2 and ProtTrans, and we managed to calculate the results of predictions for those 2 models and train them using the dataset we described before. As you can see, both models outperformed the baseline, and it looks like ProtTrans is more perspective, but we didn't have enough time actually to finish the calculations for solvent accessibility, so it's still a question for us what is going on there, but I hope that in an hour everything will be ready. Okay. So as Jen has already said, we wanted to test a little bit how different proteins are predicted depending on their size, and that's why we actually compared the different size groups depending on the protein size. Yeah, so you can see here that for the secondary protein, secondary structure prediction, both models perform similarly for different protein groups, which is actually good because it shows that we do not depend on the protein size, which is sometimes true for AlphaFold or other models which predict tertiary structure of the protein. So it's like a win for us. We also managed to calculate the results for ESM2 model and the solvent accessibility, and here you can see that for the small proteins, solvent accessibility works even better than for the larger proteins. That may be the result that it's easier to predict the solvent accessibility for the small proteins because almost all of them are like accessible. Yeah, we propose that it might be the result of this thing. Yeah, we also made an application which gives us an opportunity to insert a protein and predict the secondary structure for it and visualize it as well. So we further plan to improve it by adding the tertiary structure models there. And yeah, currently it works with 2 models, but we hope that sometimes it's possible to add another model there, like the ANG and other models. Yeah, we didn't have enough time to launch them during the hackathon, but it's still like a good thing to do further. Yeah, that's our GitHub repo, and if we want to sum up our results, then we can say that we actually managed to analyze different state-of-the-art models and compare the results for different groups of proteins, and in addition to that we implemented an application which can be used to predict secondary structure of protein and can be launched for like online if we somehow manage to deploy it. Yeah, sometimes. Yeah, so that's all for us. Thank you a lot for listening to us. We are ready to answer your questions.
I'd like to ask organizers, are we able to show the video with real-time, not real-time, but real visualization of our beautiful app work? Okay, if you'd like to see it, just come to us, make some small, beautiful speech. Okay, so would we wait? Okay. Yeah, so there we can see that we could place the protein sequence into the field for sequence, choose secondary model, choose the model of ESM, and visualize it. Yeah, so yeah, also it's able to work with FASTA files and with several proteins at one time. So thank you.
Yeah, that's the question was about the complex of proteins. Like we managed to test our model for the monomers, but did we test our model for the complex of different proteins? The answer is no, we didn't manage to do it because we didn't have enough time. Currently, we were just trying to analyze the benchmarks, and the benchmarks are usually for the monomers, like for the secondary structure prediction task. Yeah, but it's really interesting how do those models cope with complexes. It's really interesting thing to do. Thank you. It's yeah. Yeah, so the question was about that, like there are two different versions of how we can analyze the secondary structure of protein. There is like the SS3, like when there are 3 types of a residue, like that's what we did. And yeah, there is another thing, yeah, another information, like the format is called SS8. We didn't have enough time again, but we have this data, and it's like, I guess, a question of couple of hours, and it can be also tested. Yeah. Yeah, currently it's like the beta and the alpha and the loop. Yeah, so there are 3 types of residues, like 0, 1, and 2. The 0 is the alpha helix, I guess, yes, the beta strand, and the loop as well, like the SS3 format is about it.
We also have, as Elizaveta already mentioned, SS8 models, which have more than that free structure, but we just don't have enough time to visualize it on our app. But these models are also there. We also analyze them. And I hope we, in some future, do that on this application.
Yeah, before we, yeah, thank you. Before we move to the next one, just a couple of short announcements. So first, you will have a timer over there. Please follow it and do not exceed the time limit, because unfortunately, we have many presentations and we have not too much time today. That's the first one, and the second one is that I think that for the next projects we'll have to limit the conversation to, I think, 2 or 3 questions, no more than that. Again, because of the time limits. So having said that, I would like to invite the next team, and the next team is I think the SNPIC team, which worked on prediction of multifactorial phenotypes. Please, you have 5 minutes.
Repeat the question. Oh, okay, got it. Yeah. Hello, everyone. At first, we wanted to say thank you for all organizers of these Hackathon organizers. It was great event. Thank you very much that we were able to meet here, because everybody is spread around the world, and it was nice to meet everyone. So our team is SNPIC, and we worked on predicting multifactorial phenotypes, such as better breeding, and we worked on the project of Lavrenti Danilov and Mike Raiko. Yeah, and yeah. So actually, the main goal of our generation is to feed all people on the planet, and we dream big. So that's why we would like to generate enormous vegetables, and to do so, we can play wisely and use selection, smart selection. So, in general, we wanted to find a method that select SNPs, like the best combination of SNPs to predict our desired phenotype. Yeah, for this, to get closer to our goal, we used parameters of associated SNPs. We presented our SNPs as a graph, and we developed an algorithm that looks for potential SNP combinations to maximize or minimize the manifestation of the original phenotype. And we also almost wrapped it all in a convenient pipeline in our GitHub repo. Next one. Yeah, thanks. As an input, we had VCF file, like genotypes, and we used phenotypes. Particularly, we used phenotypes of soybean plant, and we considered concentration of amino acids there. We used Blink to filter our data to produce GWAS, and also we used the software called MIDESP. It's a software that finds pairs of SNP based on their mutual information. And we use this parameter later. Yeah. So here, yeah, we generated association analysis. We took subset of SNPs to consider it as some metric, as a weight of each SNP. And we constructed a graph where we defined vertices as SNPs that were taken from MIDESP, and we consider only those SNPs that had a positive beta. Edges we used, so we connect SNPs that actually are not associated to each other, and so the goal to find the best combination was to find clicks of our graph with our metric that based on weight of each vertices, such as beta coefficient, and we use some penalty and number of vertices to do this. Next. Yeah, so we can't show you a graph of unassociated SNPs, but we can show you antigraph because the graph of unassociated SNPs is a mess. So we show you this just to get an idea how does it look like. Yeah. And yeah, to understand if our algorithm works really good, that we can select effective S&P combination, we compared it with a random selection of the same amount of S&Ps, and we used the statistic metrics as a MSC and MIE, and red dots, it's our result. So it's it usually works better than randomly selection of, yeah. And we also did some annotation, and I pass the word to my colleague Vera.
So we can see in the annotation that for the first set of S&Ps, we can see mostly RNA binding, nucleotide binding proteins, and exonuclease domains. And then for the second set of S&Ps, next slide. For the second set of S&Ps, we maybe can't find any common phenotype, but we can see that there are some cell cycle proteins like choline. Okay. So that is
yeah, we have created the GitHub repo where we store our Jupyter notebooks, where we analyzed the MIDESP data, and also there is a Python package that which is basically a pipeline that does the initial steps like peeling, filtering, and MIDESP. Yeah, thank you for your attention.
So the question was if we split the data set before to train to test before select, yeah, so for everybody, I repeat the question. So did we divide our data set into train and test before selecting SMPs, right? Yeah, so selecting randomly selection, do you mean? Yeah. Mm-hmm. Okay. No, we found the clicks and the best combinations before splitting trend and test data. How did we find the clicks? Can you use? Huh? So yeah, the algorithm was it was just like based on clicks, on the weights in those clicks, and we selected the best through the whole set of clicks. So it's not like some common.
Yes, we have had it. So we developed an algorithm that can define the clicks, and so we can also find it manually looking on the plot. Algorithm, I can't name it. We just wrote a Python script. We just wrote a Python script based on the data. We didn't use any algorithms like maybe DFS and so on.
Thank you. Yeah, next up is a team that worked on new aging biomarkers. I think that don't have a name on the slide, but still, yeah, please, the floor is yours.
Yeah, thank you. So in the next 5 minutes, I'll try to tell you about biomarkers of aging and what our team tried to do with them. So, yeah. The first thing is, what are biomarkers of aging? These are some biological parameters which can predict age-related changes and age-related disease. Sorry. Our goal was to analyze RNA sequence of patients with different age to find genes expressions that can serve as predictive biomarkers for age and age-related disease. Our study consisted of 5 different parts: raw data pre-processing, data processing, differential analysis, machine learning, and the last one, function analysis. We studied the cohort of 133 cell lines derived from 133 healthy individuals and 10 patients with progeny premature aging disease. I have real problems with this presentation, sorry. So the first part of our results is differential expression analysis. It was studied on healthy and diseased patients. We found out that 6 genes with increased expression levels among diseased samples differed from healthy individuals, and 3 genes with significantly decreased expressions also differed from healthy individuals. Here you can see a heat map. And we tried to find a connection between these genes and pathways of aging and cell longevity. Here you can see the pathway analysis based on Go enrichment, and this bar plot shows that identified genes are significantly associated with following mechanisms. And the next one, we try to find connections between these pathways, and as you can see, the majority of genes correlate with each other. For example, genes involved in tube development are connected with those ones participating in tube morphogenesis and responsible for cell motility. And these are all genes that we studied. And the second part of our analysis is biomarkers that we found out during our machine learning analysis. Here are 11 genes that we found. We tried to connect those gene expressions with ages. We found 4 groups that correlated with the gene expressions. And we tried to take 5 genes from these 11 that we considered a biomarker for aging. First 3 genes are connected with heart disease and overall cardiac function. It's SRA, RPL3, and CREAM1. And as you can see, there is a peak on the second age group, which is considered a group from 40 to 50 years. The next biomarker is TNC, and this biomarker is an element of extracellular matrix, and these results also show us the connection with matrix and glycosylation. Here you can see a biomarker ADPGK, which is an ADP-dependent glucokinase. And the last biomarker is biomarker related to osteoporosis and osteogenesis. It is we want to believe that this biomarker can predict aging because of its connection to osteoporosis and similar diseases. And our conclusions. We found genes that connected with cellular matrix, and they were significant during our differential analysis. Also, we found out that control samples of 40 years old possess significant increase in expression in biomarkers associated with myocyte skeleton. Gene enrichment levels that significantly express genes are associated with epithelial to mesenchymal transition in colorectal cancer, prostate cancer, and gastric cancer. And 2 genes with increased differential expression participate in glycosylation of cellular matrix. And these findings correspond with modern theory of aging about the glycosylation of cellular matrix, and also add on to existing biomarker databases, which we studied during our research. Thank you for your attention.
I have several comments about our project. Firstly, we've seen yes. Okay. Just 2 sentences. We've seen a transition, a transcriptomic transition that means yes. I'm a team leader. Okay, we've seen transcriptomic transition at about 40 years. That means that at 40 years transcription become different, and it may be linked to menopause, linked to imbalance of men hormones, and also about linked to insulin resistance, which is part of modern theory of aging. And no, so that's all. I want to say that our work is kind of revolutionary because of because no one made such algorithm for aging research. So we have a QR code. You can look at our data. There are our contacts. So please contact us if you have some questions.
Okay, thank you. Yeah, we have, I think, time for one question from the audience. But only one, yeah.
So we've shown changes in expression of genes that were seen in differential analysis, and the question was, do we see the similar genes, the exact same genes in our machine learning analysis? So yes, we've seen, and in the conclusion there were 2 genes that related to extracellular matrix, and they were seen in healthy. It was elevated in healthy and disease patients, yes. No, we haven't. We didn't have time.
Yeah, again, a short announcement. Project supervisors are not supposed to stay here with the teams and comment on the work, so please, for all the project supervisors, please do not come on stage with the teams. Yeah, next up is the team that worked on antigen, antigen detection and annotation. Please, your 5 minutes. Please mind the timer, and I will stand up 30 seconds before you have to stop the talk.
Hi everyone, our team name is Transpozov, and we're happy to perform you the results of our study dedicated to automating the detection and annotation of all antigen operands in prokaryotic genomes. All antigens are highly variable structures located on surface of mostly gram-negative bacteria, and they have been recently widely studied due to glycoconjugates vaccine design. Hence, it's extremely important to identify and locate all antigen operands in prokaryotic genomes. During the time we worked on our projects, we concentrated on following tasks. We explored all antigen operands of model species such as Escherichia coli, Salmonella, Enterica, etc. We collected the database of known all antigen genes. We came up with approaches and chose the best tools to combine them all in the pipeline. So now we're happy to show you WOOF, our wonderful all antigen operands finder tool. Here you can see its workflow. The user is required to upload genome assembly, and as a result, the user can get all antigen operands annotation and the graphical reports. The user can also upload transcriptome and /or reference annotation in order to get even more useful information due to annotation check and validation. So,
yeah, the workflow is very complex, but the usage is rather simpler. You can get our tool with GitHub, and you need to prepare just one file, your annotation, a genome of your favorite bacteria species, and then you just need to run only one command of snakemake, and that's it. After 20 minutes, approximately, you will get the results. And does it worth to wait 20 minutes? Definitely yes. We have 3 types of the results that you can get. First of all, you will get the list of all antigen operands in this genome. Also, you'll get annotation of all the genes located within these operands. And also, you'll get the nice representation, illustrations of the operands. What's illustrations? It looks like that, several pictures for several operands. You get like unique operand ID, the genes located within the operand, and also you will have some functional annotation, some text to take a closer look at what you've got. But you may ask, hey, for the bacterial genomes, you may have different problems with mobile genetic elements, and that's it. And we've prepared a special model to deal with transposons, because transposons can spoil operand structure, and we cut them from the genome, then we call operands, and after that we will add them back again to visualize with red dangerous color these bad guys. And finally, you may say that that's it, that's results, what's interesting, so on. Here I want to illustrate really wonderful results. For example, here is the real Salmonella enterica O antigen operand from some paper that was defined with wet lab methods such as PCR, and we found this particular operand with our tool. For some reason it was split into 2 parts, but what's really interesting, take a closer look at this red gene. It's inverted because it's located on the negative strand of the DNA. However, with the paper, we see only one gene downstream of this selected gene, and here we see the wide range of genes found with our tool, and not all of them are annotated. And this is really beautiful. This is a way not to look only under the lamp. With these genes, we can take a better comprehension of operands, of their structure, and also we can study better the all antigens, the genes that involved in the biosynthesis and biochemistry. So you can check out our GitHub repository, and we'll be happy to answer your questions. Thank you. This is a really good question of validation. When we started to do so, there was a question that we found some operands, but do we compare with the existing well-known operands? And yeah, we tried to compare with some of them. For one, we found interesting examples, but the task to compare all the known operands with our tool is the task for the future.
Well, actually, we compared the results of our tool with the results of known Salmonella enterica annotation, and we found 82 or antigen operon genes located in different or antigen operons, and only one gene we missed according to the reference annotation.
There was a question about overlapping operons in the bacteria. Yes, this is a really big problem, and
Yes, thank you for your question. It was out of scope of our research. Yes, because we took already existing tool, and these tools are focused on the search of these operants. So probably no. Yeah, also we found some overlapping genes. Actually, transposons was these overlapping genes. So kind of, not all cases, but some of them.
Thank you very much. Yeah, please stay on time like this team. They have the clicker. Yeah, thank you. The next up is a team that works on cell-free DNA in oncological patients. Please, your 5 minutes.
Thank you. My name is Mariana. Let me introduce our teamwork results, and I hope it works. Oh, yeah, it works. We know that liquid biopsy is a very promising tool for precise oncology, but it needs development of the robust pipelines for the data analysis. So our goal was to try to develop such a pipeline, and we have to filter the false positive events, and to analyze the data and create the pipeline, and try to interpret the results. This is our data set. It was provided by Boston Gene and contains the data of target deep sequencing for 4 patients and 5 time points for every patient. And every point has 2 replicates, 2 technical replicates. And you can see that all the patients are different on their diagnosis, treatment, and clinical stage of the disease. And patient 1 and patient 3 have a visible change in their clinical stage during the observation. Okay, this is our proposed pipeline. We have VCF files as input files and some kind of another files as output. And we designed a tool for oncology traces tracking, and it has a name. So briefly, first of all, we need to filter the data to remove false positive events from them. And we have some modes of filtration: soft mode, medium, and hard, and some special filter for clustering. Then we have like 3 branch of our pipeline. The simplest is just to annotate the filtered variants. Then we do clusterization to find the genes which time changes are correlated, and annotate them too. And the magic branch of our pipeline is to make PCA to track every patient state on time. This is an example of the simplest, excuse me, thank you. This is an example of the simplest way to use our pipeline, just to annotate the filtered variants. And this is a piece of annotation of further analysis of annotated variants for just one of the patient. And you can see here the graph of interaction of the protein affected by found variants, and a piece of annotation with gene ontology database, KEGG database, and there are a lot of databases about these gene functions and gene pathways, etc. So this data can be further analyzed by biomedical specialists if it needed. Thank you. The next step, the next idea is try to do some correlations. So we thought that the changes of variant allele frequency through the time can represent the changes in tumor condition. And then we do some correlation analysis to find gene which whose changes are highly correlated. So we can think that these genes maybe come from tumor together, etc. And there is an example of such cluster analysis, and there are 4 clusters on the picture, and inside each clusters there are variants which whose changes are highly correlated. And on the right side of the slide there are some details when we jump from variant to gene affected by this variant. And this is our PCA analysis to track the each patient data. And if you can see the blue points for first patient, and you can see that first time points are different for the next 3 points, and it shows the change from remission to relapse. Yes, I want. We have working tool. We are very proud. And this is our GitHub. Please ask question if you want to know something. If I understand correctly, the question was about filtration on the depth sequencing. Yeah.
Yeah.
So the question is about pre-filtering the data according to the depth of sequencing before performing the analysis. And we were provided with the filtered data, so we didn't have to do all the pre-filtering stuff.
Thank you.
Now we move to the biostatistics part. So yeah.
Yes, and I'm happy to announce our next speakers who will represent the project devoted to retrospective analysis of complex disease trajectories. Thank you.
Well, thank you very much. Our team is called HemaHazard because we have both from hematology and from statistics in our team. Where is so sometimes the complications of our treatment, like graft versus host disease, can become the reason of the death of the patient, not the acute leukemia, for example. So the first challenging part was creating data frame. As you can see, it's a hell for all data analysts here. It took us a lot of time. Making the same format of the data and connect data to itself, and also regular expressions to combine this data frame by IDs. So when we explored our data, our primary outcomes was overall survival and event-free survival, secondary severity of acute graft versus host disease. But we also have limitations. There was only 333 patients. All these patients experienced either acute GVHD or chronic GVHD, and no data about prior disease status. So we made a descriptive part. Here's the first finding: haploidentical transplantation increases risk of death in our population. Then at the point of interest of acute graft versus host disease, it's a time-dependable variable. We provided landmark analysis based on literature data, so it's also severe GVHD decrease probability of survival in our patients. Then we provided time-dependent Cox regression, where we found that acute GVHD as a time-dependable variable has an influence of, and male gender has an influence on overall survival. This is comparable with literature data. However, when we used chronic GVHD as a time-dependable variable, we found that something unusual, that it is CMP, it's chronic myeloid neoplasms, has significant influence. And this is because this data has only patients who engrafted. Patients who has no engraftment, usually patients with CMP dies from from graft failure or graft rejection. So the same is similar for event-free survival, and this correlates too. So then we come to machine learning part. First, we decided to try to predict severity of acute GVHD in our sample. Here you can see using decision tree model. Here you see that two confusion matrices. They give us false positive results, as you can see on the matrix, but for practitioners, from medical practitioners, it's better to find a patient who will probably experience acute GVHD and pay attention to him, than not to pay attention at all. Then we tried to predict overall survival using classical machine learning algorithms. So here you can see that results were unsatisfactory, and then we tried to use scikit survival package, and we applied two models, random forest to survive and gradient boosted models, and we received very and the time-dependent area under curve was almost 0.9. So after this, we made feature importance analysis, and we found that in this model, GVHD type, transplant type, have very big importance in prediction of survival. So, in conclusion, acute GVHD, gender, type of transplantations are the potential risk factors for overall survival and event-free survival. Decision tree models could highlight patients at risk of severe acute GVHD. A specific survival machine learning algorithms could potentially improve decision-making for inpatient management. Of course, we need to use bigger data sets to confirm these models. Thank you. Of course, no. I think you speak about this model, yes? To basic statistics, you mean? Basic statistics, we used GVHD as a time-dependent covariate, so we don't know, of course, for Cox regression models. It's a time-dependable variable. And for this, and for this model, can you help me?
Okay, so I think what you mean is, at the moment of transplantation, we do not know if the patient has acute GVHD or not. The study cohort was composed retrospectively. So those who have developed acute GVHD after transplantation, yes. And due to the fact that we start the point 0 is date of transplantation, but a GVHD happens after that, we included that variable in the model as a time-dependent variable.
This is epidemiological trial, so it's like the real situation, like all patients come to the study and we research them. Are you asking about the like amount of factors? Do you mean like there are too much factors? Usually we have like a rule that we can include one risk factor to 10 events. So we used this rule, and that was like factors that we have chosen. Like we have about almost 90 events in overall survival and a little bit more in event-free survival. So it's like 1, 2, 3, 4, 5, 6, 7, 8. Yes.
And the next team tried to apply natural language processing methods for parsing histological and endoscopic studies results.
Good evening, everyone. The name of our team is Fix, and the and the project we will work on the project dedicated to application of NLP methods in histological and endoscopic studies results in oncohematological patients. And the main aim of our project is determine key features in the text description of histological and endoscopic studies that predict clinical diagnosis as graft versus host disease, and it is and we focused on solving this problem mainly. I remember you that graft versus host disease is very serious and frequent and very serious complication occurring after allogeneic hematopoietic stem cell transplantation, and substrate of this complication is immune attack transplant cell to host cell and following destruction. So solving our problem, we met some problem, including a small data set, specific medical terms, and labels in description. And we analyzed 85 description, biopsy description of gastrointestinal tract. The example of histological description is presented on the slide. This text is very hard for understanding and contain many specific stylistic features and sometimes mistakes. And for our analysis, we used TD-IDF method for making clusters and n-grams. This is our n-grams. There are 4,577 n-grams. It is big data, and after it we reduced these n-grams which not containing some important information in road mode, and after it we used a different classification methods, and the classical method of classification as boosting provided us 27 key features, and it have more stronger ability to predictive of diagnosis graft versus host disease. So our conclusion are presented on the slide. We have identified 27 key features from 2,577 after classification, but not all of them have really clinical significance. And in it we may make decision support system helping doctors in biopsy analysis. Thank you for your attention.
Thank you for question. It may be utilized with entity recognition. Выделение сущностей. Entity named. Скажите, пожалуйста. NLP classical task like entity named selection, like something like this. Yeah, distinguish. This is a more important point. Yeah, it is. Yeah, it's a good question. Our idea was about learning birds pre-trained, then
train a little bit because of a little new words, essential part. And then one perceptron layer, because we can't learn much parameters because only less than 100 persons. And it was not okay because also attention layer, we should select key features. We don't need, we need a high score, but essential point is key features, and this matrix is from latent representation to labels. It's no sense for us. We can distinguish, and we should train attention layer mask, attention mask, and we can't do it with 100, unfortunately.
So I would like to one more repeat the information. So first of all, we haven't got a second microphone, so please repeat all questions to you in the microphone so that people on the translation can understand on which question you answer. The second one, we have a clicker, and the acceptable one of clicker is on that laptop. So if you would like to move on the presentation, please move it to that, not to that laptop, and move it by the up. The up is smaller, the down. And if you would like to move by the presentation, make it slowly and carefully, please. It's tried. So, thank you, and we would like to see the next comment.
Yes. Now it's time to get something new about Q receptor, please.
Okay. Hello, we are the Natural Killers team, and the subject of this weekend for us was evaluation of KIR impact on graft failure after allogeneic hematopoietic stem cell transplantation. Okay, graft failure is a group of complications after allogeneic hematopoietic cell transplantation, which is a method of treatment of different blood system diseases. The markers of it are yet, we need more diagnostic markers to prevent and possibly, we need more diagnostic markers to predict and if possible to prevent that complication. One of the possible markers is KIR. What is KIR? It's killer cells immunoglobulin-like receptors. They are located on natural killer cells, which are the first cells to recover after stem cell transplantation, and several cells, natural killer cells, are also detected from the host as well. It's a chimera, actually. KIRs regulate immune response by interacting with HLA, but still the influence of KIRs on graft failure is yet to be found. There are so limited data. Which were our tasks? You can read our tasks for the weekend here. Let's go here. Our data set, our primary data set, has included 118 patients, but we excluded patients with other diagnoses than acute leukemia. We excluded patients with relapse and with myeloablative conditioning. Our following calculations were performed on 69 patients. We found some correlations between different Q receptors. The red light, the red color stands for a positive correlation. That means that these subtypes of KIR receptors are more likely to be found together. The blue color surely stands for negative correlation. Whew. We also found that the mismatching in graft versus host directions are more likely to be found in transplants of related donors, which has also been shown to have some protective effect. Okay, we have also researched some factors connected to graft hyperfunction, including the KIR type, HLA, and different mismatching types, and we found some significant ones. For example, the age. The first one being age. The older is the patient, the more likely is graft failure development. The second one is the transplant type. I mean, the bone marrows are more likely to fail in comparison to peripheral blood stem cells. But there are some protective factors too. For example, the 2DS4 subtype of KR. And the second protective factor we found was a mismatch in the graft versus host direction in subtype HLA A0311. We also built a multi-factor prediction model which has showed better and more exact results in predicting than each of those factors separately. We can use this model to predict graft failure, but the model is limited due to small sample size and it needs more external and internal validation. Yeah, the last task we have also completed. That was really fun. And the conclusions are written on this page. And yeah, surely the last one. Yeah, that's it for today. And if you have some questions, feel free to ask. Sure. Yep. What? The main results. Okay, we found a correlation between KRR receptors 2DSD2. There's a positive correlation. So we may probably, yeah, this slide would show better. The red ones is the correlation, the positive correlation. The strongest one is between 2DL2 and 2DS2. A strong correlation is also to be seen in 2DL5 and 2DS1, 3DS1. The input data was the analysis of KIRs in hosts, in patients.
No, it just was the information about presence of the receptor. We received 1 or 0 for the presence or absence of receptors on the cells. No, just have these receptors or do not have these receptors. And so on the this slide we can see which receptors are more likely to be emitted together and which are more likely to be absent. Yes. And so after that we just performed many univariate regression models, took the significant factors, analyzed it more precisely, for example, age, age, transplant type, one of the receptors shown on the previous slides, and mismatching in the receptors. And after that, we united these significant factors into the one logistic regression model, and this model allowed us to make more precise prediction than the predictors separately. So about the limitation, we also talked. Да, я понимаю. Yes, we made our dataset consistent to the diagnosis, so we took only acute leukemia, so the general population for which we propose our conclusions is only this diagnosis, and other factors we also tried to minimize the variance, because we excluded severe type of myeloablative conditioning, and no. Only acute leukemia, yes. Yes.
Yes, and so now we are moving towards oxological predictors of fetal growth, led by Dmitry Zhakota. Thank you.
Thanks. Does it work? Like that. I see. So, hello everyone. We're the team body of evidence, and the project was dedicated to oxological predictors of intrauterine fetal growth restriction, which is basically the undergrowth and underweight at the respective gestation stage compared to overall population. And the conditions basically start but at least 3 different medical, clinical disciplines which do not agree on their approaches or even the definition of FGR itself. So the data might be even quite hard to commensurate between those disciplines. So the project was about, first, to estimate whether the data from obstetrics, neonatologists, and pathologists can be compared uniformly. Second, whether these data actually contain any interesting patterns for FGR development. And third, whether we can leverage and improve the known FGR predictive criteria. So, the primary data set for this project was the autopsy data for 3,300 stillbirth, postnatal death, or miscarriage cases, with each case bearing the gender, gestation stage, and some morphometric parameters, and sometimes also the clinical FGR diagnosis. The problem is, while obstetricians and child healthcare doctors basically work with normal children, the material for the pathologist oftentimes looks like that. The problem is the data is heavily affected by the pathologist, resulting in the distortion of morphology, and therefore the data had to be severely cleansed of missing data and outliers, and also those values who were imputed. So, which is represented by here, especially, for example, for placental or pancreas. So the first task was to compare the pathological data to published data from papers or from the Intergrowth 21 consortium, which is basically about ultrasound assays. The problem is oftentimes the data is presented not as raw data, but as the selected quantiles, such as percentiles, and therefore it is very hard to compare to our data. Nevertheless, with certain impedance techniques, we managed to reconstruct the missing gestational stages, missing quantiles, and so on. And where it was possible, for example, for the internal organ mass, for the other pathological assays, we also managed to reconstruct it by our data with a plausible level of confidence. So once we did that, we moved to the next step, whether we can dissect any interesting patterns in these data, and we decided to go to two long-running assumptions in the neonatal, in the oxology. The first is the impact of gender of oxology and FGR. Apparently, the data does not differ much, at least for the initial glance, for female, male, and combined data sets. So we first performed the t-test pairwise, gestation-wise, and then went deeper for our oxological pathological data set for different characteristics such as internal organ mass, body weight, and apparently these do not really differ much. So our verdict most likely doesn't differ much, and it's basically running traditional oxology. The second question was the assumption that pathological data oftentimes underestimates the weights compared to ultrasound assays. As you can see here, the red line is a bit lower, but not statistically different. With the t-test, you get somewhat significant results, for example, for the combined female and male data set, but once you adjust it, no. And the same goes for example, umbrella meta-analysis tests and umbrella meta-analysis tests and one sample mean meta-analysis tests. But once you, for example, treat the data as a real time series, with the CHO test, for example, you really get a certain difference in trends between ultrasound, pathology, and neonatology in terms of pathology underestimating the weight. So our conclusion here is that needs certain evidence, quite inconclusive still, but definitely worth digging deeper. Sorry, I'll be fast. So the last question is that we try to leverage the, try to predict the FGR condition by the anatomic data. The problem is oftentimes the FGR is neglected by the neonatologist who passed the material to the pathologist. So the data set was underestimated in terms of FGR cases. So we added certain FGR cases judged by the two known and widespread characteristics or criteria, if those agreed, which resulted in adding more than 100 more cases to the data set. And those were used as labels for classification with extreme threat boost. The stats are most for bragging here. What I want to draw your attention to is that basically, first, the gender is still not a very important feature in terms of feature importance. And second, some elaborate morphological criteria, such as, for example, body surface area and its modified versions or ponderous index, are quite informative. Even if you take only those, the model doesn't lose much in terms of, for example, its precision and accuracy. So our main conclusions is that oxological observations from different sources can be compared. They are comparable, at least in terms of size and weight. Second, the several assumed patterns from the field were dissected, and apparently they are quite inconclusive. Maybe because of how data is presented in the literature and public databases. And third, advanced toxicological indices and morphological indices may serve as plausible FGR predictors based on morphological and pathological data. So, we would like to thank the organizers, the committee, the venue holders, our mentor, the coffee shop next door, and finally you for your attention. Which is this one?
Yes, this one. So how do you see the application in clinical practice of this particular model?
Well, think of it this way. The pathologist has a very direct, the most direct possible access to different morphological properties of a child. On the other hand, to save the child from the aggravated conditions caused by FGR, you definitely wouldn't like to dissect it. So once you, for example, find how those can be applied to the ultrasound or any other data from obstetrics, and then you can infer a model from this, so the indirect link can give you a right level of, I don't know, confidence in how these data then be further applied to ultrasound and the actual living fetuses. Beg your pardon? That's basically on the overall set, but the model itself was tested like with the stratified k-fold 10 times.
And the last but not the least, before the break, is Trip to Fun project.
Good afternoon, everyone. We are Tryptophan, and today we are analyzing T-cell immunological landscape in its association with graft versus host disease, particularly after bone marrow transplantation. We were provided with the following data. We had 67 patients with leukosis who underwent bone marrow transplantation. Some of these patients developed chronic graft versus host disease. Blood samples were collected at multiple time points after the transplantation for all these 67 patients, and they were analyzed by flow cytometry. Particularly, T cells were analyzed. For each T cell, surface markers were analyzed, maturity, and cell type. Totally, we had 154 combinations of all these markers. There's important note here that some of these populations might intersect. So the first thing we have done is we have removed fully duplicated T-cell populations, and we have also removed T-cell populations which possessed abnormally high or low dispersion. Totally 24 cell populations were excluded from further analysis. Then we have tested each and every individual T-cell population to see if they are associated with GVHD onset. Samples were analyzed only from day 90, as this is the most challenging task. On this state, we had only 3 somewhat significant results. Only CD8 cells with HLA-DR expression and T helpers possessed some significant ability to distinguish these patients. We have also performed Cox proportional hazard univariate analysis, and we have obtained pretty alike results. We have observed that T helpers and CD4 cells with T maturity are associated with GVHD onset and higher levels of CD8 cells with different stages, but mainly activated T cells who possess CD226. For example, they are associated with the lack of GVHD or its late onset. We have also taken all potentially interesting features and tried to apply a bit complicated models, incorporating all the aforementioned features. We have started with clusterings, and no matter which clustering approach we tried, we got pretty much consistent results. Our patients were split into 2 major clusters, which significantly distinguished patients who had GVHD onset and those who didn't. These clusters were always defined by CD8 active cells and CD4 naive cells. You can also see they are clearly distinguishable on our UMAP plot. Finally, we have decided to put some ML models to the test. We have taken the most popular classification models, and we have calculated the most popular classification metrics for them. We have performed this as well on the whole data on day 90 using threefold cross-validation. So, these all metrics are the mean from these threefold cross-validations. And despite the lack of data, I remind you, we have only 67 patients, we still get somewhat significant results, which of course are to be refined as long as more data is collected. Here, the potential leader now is a random forest classifier. Finally, we have taken this random forest classifier, And we have plotted feature importances using the SHAP package. See if we get consistent results. I mean, if we get results consistent to our previous findings. And it turns out like pretty much yes, we get I guess pretty much consistent results. I'd say they're almost identical. So our main conclusions are the following: that CD8 T cells of any maturity but predominantly active are associated with better outcomes after bone marrow transplantation, and CD4 T cells, mainly low effector function, are associated with chronic GVHD onset. We would like to thank Michael Drokov for providing us with data, our organizers for providing us the possibility to participate in this hackathon, and now we are ready to answer your questions. Sorry, could you repeat it? Yep, I try. Oh, you mean this one? Well, there's pretty much to to mention, because originally we have 154 populations and we removed 24. So as far as I remember, we predominantly remove some white populations. For example, we had results from this flow cytometry, something like the following: CD8 plus T cells with unknown maturity and unknown expression markers. They are obviously include some various populations which behave differently. And as far as I remember, these were the major populations that were excluded due to their extreme variability. No. Yes, all these populations were present in each and every sample. Oh, as far as I remember, it's somewhat even. Some around 30 in each group. No, we are trying to create some prognostic models because, on day 90, none of these patients developed GVHD. They have developed it later, and sometimes we have the exact day of the GVHD onset, and sometimes we have, for example,
No, some patients were censored because they were still alive at the last day of the follow-up, which were the blood collecting days. So 19 days, it was after the allograft transplantation, and the 19th day was the first of the blood collection. So here all of the plot starts at 90 days because we are interested in the CVHD prediction. Well, as you see, when the curve, when it goes up, here we have an opposite of the survival, or not survival, but of the probability of not developing. So here we have a risk. So each time the curve goes up, this is the patient. So the first one was on the 140 days or even later. So 19 plus 50. Yeah, there were only chronic GVHD, yeah.
Thank you. And it's time to make a short break. 10 minutes break to relax, not relapse. So.
So for everyone to remember, we meet here at 10 minutes to 6 p.m.
it's time to come back because we have to listen another 10 projects. Okay, we need a few minutes. Guys, please see the chat. There are any announcement about your stuff in the other hall in Tarson. If you have some in this hall, please take it. And the next project, it's Euros project. It's the team, the root of evil.
Thank you. Hello, everyone. 2 days ago, there was an interesting lecture. Trying to. Can you help me with this, please? Good afternoon, everyone. So 2 days there was a great presentation, which it was talking about the dark and the light side, so the biostatistics and bioinformatics. So our presentation is devoted to the root of evil, but we are concentrated on the microbiome. There was an interesting Nature Communications paper back in 2020, where based on the human microbiome composition, the authors managed to make a mathematical model that actually distinguishes between healthy people and non-healthy people, whatever the disease they have. And it turned out to have quite a good accuracy of 73%. So this was like a meta-analysis of a number of human studies with more than 4,000 participants, including 12 different diseases. Okay, and so in this study, what they did, they made a list of the health prevalent bacteria, also good bacteria as we can simplify it, and health scarce bacteria, the bad or the evil bacteria. So there were 7 good bacteria and 41 bad bacteria. And actually, the aim that we had, the aim of our work was to understand what is the difference between the good and the bad bacteria. How can we differ the health scarce bacteria from the others? Okay, so the first thing we wanted to check is, of course, to check the taxonomy. So are the health scarce bacteria grouped somehow in some taxonomic categories or not? And here on the left-hand side of the slide, you can see the health prevalent bacteria in blue and the you can see the health prevalent bacteria in yellow, and you can see the good bacteria in blue. So there are 7 good bacteria and 41 bad bacteria, and there is no obvious clusterization of the bacteria in any of the taxonomic categories. Okay, and so next we were applying bioinformatic methods to that, and first of all, we were applying methods to the whole proteome, and please, Olga. She will tell about
that. The idea was to look for amino acids chimeras that are situated with metabolic or immunogenic activity. We created the data set with chimeras frequency, and when use random forest classifier, so our scores aren't so awful, but when we try to define meaningful features, and where are a lot of features in with data set, we haven't found them. So maybe we need to recreate the data set to make chimeras longer, but we need more computational resources for that.
And then our next approach was concentrating on the genes that we see in this bacteria and finding any functional overrepresentation within these genes. And so we studied the gene ontology terms. The analysis was done on 7 health prevalent bacteria, and also we added 21 neutral bacteria, just chosen from the ones studied in this Nature Communications paper, and it was compared to the 41 health scarce bacteria. So the bacterial genes were assigned, the gene ontology terms were assigned to these genes. A total of more than 2,000 gene ontology terms was there. And then to see the difference between the good and bad bacteria, we used the Wilcoxon rank sum test. And then we're correcting the p-values multiple testing. And overall, we had 26 results, 26 Go terms that are significantly enriched in either of the groups after the multiple testing correction. And so let's see them. Okay. So as you can see that out of these are just 5 top hits, and the corrected p-value for each of them is less than 0.002. So 3 out of these 5 hits, they are related to transmembrane transporter activity as amino acid or nucleotide. And also there is a sulfur compound metabolic process. So sulfur compound metabolic process is underrepresented in the bad bacteria, and the transporter activity is overrepresented in the bad bacteria. Here is the same data, just presented in a bit different way with the exact p-values after corrections that you can see. Okay, and now Grisha, please.
Okay. Also, we run some additional pipelines, and on the general portrait pipelines, we find that they differ much, but we not find any interesting. It's not very interesting. We don't have time, but we tried a anti-smash pipeline for finding some synthetic gene clusters, and we found a lot of differences between ill and good bacteria. And most interesting part of this is that we found that the good bacteria contains more substituted clusters than evil bacteria. Also, we tried another pipeline for finding IS elements, but we didn't find nothing interesting.
Okay, so the question is about the colors of good and the bad bacteria. That might be a bit confusing, so I'll say it again. So the bad bacteria are are in yellow. The good bacteria are in blue. There are not a lot of them, just 7. And the others, the black ones, are just the other bacteria, neither good nor bad.
So maybe it's not clear from your distance. Here we have colors for phylas, like pink is Proteobacteria, but species names are also colored with blue or yellow. So yeah. So there is no correlation with phyla at all. Yeah.
Yes? Yeah. Mm-hmm. No, we didn't find any feature that is. I don't think we found anything that is common to all of them. It's just about the overrepresentation. So that's just a statistical correlation. Not all 41 of them. Okay, thanks for the question. I think that's a good one. Actually, we ran all the bacteria as a whole group. We didn't group them by families. So this result was obtained just when we were taking 41 bacteria just as individuals.
Excuse me, do you have a clicker? Okay, clicker is here, I see. Okay, yeah, next up is the team that worked on the Sex of the Brain project. So please, the floor is yours.
Что происходит? Здравствуйте. Ladies, gentlemen, and non-binary kittens. And first of all, I need to say that this is the wrong slide, and it doesn't work? Yep, this is the right one. And let me introduce our team. It's Alexandra, Anna, Savili, and the lead of our team is Ilya. It's magic. Okay. The cheap and effective method to study brain activity is EEG. A lot of EEG data were collected and stored in open and private databases. Descriptive statistics of EEG can be effectively used for classical machine learning approach. But also we have a lot of raw data. And we can use this raw data to try to develop something with these neural networks and extract interesting insights. And for example, we can use it for predictive personalized medicine. Previous studies and our results demonstrate that although there are sex differences in brain structure, brains do not fall into two classes. One typical of male's brain into two classes, one typical of males and the other typical of females. Real data is more complex and more difficult to analyze. Perhaps male brain, female brain data is continuum.
Okay, now let's move on to the modeling part of the project, please. Let's talk about feature engineering. Applied machine learning is mostly about feature engineering and other types of data wrangling like cleaning, transforming, and we were doing those kind of things for the most of the time. We spent a lot of time doing literature review and experimenting with different features to understand which are better suited for our task, and it turns out that the best way to represent the complex EEG data in a simple design metrics is to use different types of waves, use waves of different frequencies. Those types are denoted by Greek letters, and they are correlated with different types of behavior. For example, gamma waves are correlated with problem solving and concentration, while beta are correlated with busy active mind and also empathy. So we decided that it's going to help us solve our task. Now let's move on to some results. Here we compare our results with the state-of-the-art tools. Two right columns are our results. We used two different approaches. First one is to use a compact convolutional neural network on raw data, and second is to use our features, less data, less subjects, and use gradient boosting on decision trees, which is a bit more interpretable. By the time of presentation uploading, our scores were 69 and 64 for balanced accuracy, but just after the presentation upload, one of our models finished training, one of our deep learning models finished training after long hours, and we managed to achieve a score of 79.8, which is just about the state-of-the-art score. So let's draw some conclusions. Firstly, we found it really hard to accurately predict sex based on EEG data. We used some unsupervised methods, but they failed completely. They managed to capture other kinds of differences like age, but not sex. When we used our supervised models, results ranged all the way from 64 to about 80 for the best model. Maybe it's just about the limit for this task, but we have some ideas to improve the score. What do we need? We need more data, more time, more computational resources. Why do we need it? To do the optimization of our best model, to construct new features, and to extract data from other types of sources. And I would also like to ask Sasha to add a couple of words about the best model.
I think it is a quite ordinary convolution. Yes, but maybe about tuning, it is, I want to add some layers, and maybe when I explore more about custom datasets in this PyTorch framework, but not PyTorch in a classic way, I think I improve the results with transformers or other networks, not only convolution.
And also, we have, I'm sorry, 30 seconds. Yeah, we have a, I'm sorry, but we have the best model for predicting sex by brain activity only and using only one feature. This feature is, do you like pizzas with pineapples or not? But outliers happened.
Yeah, thank you for your question. Do I need to repeat the question? Yeah. So the question was, did we use not only time-dependent characteristics, but also like frequency ones or others? So we used not raw time, but we generated epochs from these times, and we just I think removed some of the data by these and generated less memory consuming data, but we didn't try to I think to change time to something else. So we always worked, we always worked with times and with channels. Sometimes we just changed the channels or just extracted features from these channels, but always with time. Yes. Yeah, thank you. We analyzed those data sets separately, so they are different in terms of channel sizes, electrodes numbers, and also I think in metadata. So we didn't merge them, just analyzed separately.
We tried to use unsupervised approach, but did it by classical machine learning approaches, and no interesting info from this approach.
Yeah, but the question is good in terms of that there is a discussion about do we need to classify brains at all and sex-based. But yeah, it may be interesting in terms of diagnostics for some diseases. Maybe, we don't know yet. And there is like two different theories, this continuum one and like a distinct one. So.
Yeah, we are already over the time, I think, for half an hour, so that's not good, I should say. Okay, next up is the team, I think, Zorawar, which worked with the gene company number.
We've had Barbie version, but where is Oppenheimer's version? Okay, so we're a Team Zorawar team. And our project is determining the number of copies of specific genes in a tumor. I'm sorry. Okay. Thank you. What are copy number alterations and why are they important, especially in tumors? Mostly because some therapy schemes strongly rely on number of copies of the genes, of some certain genes, and those kinds of alterations can interfere with current therapy a lot. So why is there a problem? You can see the real data that we got from our supervisor. So we have one patient, but and 2 biopsies, and they get like completely different results. Some of them say that there is no duplication. Some of them say that there is gene loss. We've had a plan and we stick to it. So our major goal was to perform some tool that could be better than existing tools. There are actually two. Yeah, there are actually 2 major waves of copy number alteration calling. First of them is alternative allele frequency that strongly relies on heterozygosity of the organism, but sometimes it cannot be obtained for a certain gene. As I say, we actually are looking for some focal events inside of one gene, and sometimes we cannot obtain heterozygosity for a gene. But it's a legit way to determine Yes, and the other approach is depth ratio. It's based on coverage from NGS data, because mostly NGS data is used for this type of analysis in clinical practice. So we combined an output from an existing package SNV kit and also perform some custom estimations with the smaller bins to actually track focal events. We also used a BAF approach, and our results are as following. As I've mentioned before, BAF sometimes cannot be estimated for a certain gene, and our depth ratio and results from CNV kit, they align quite a lot together. Unfortunately, due to the lack of the time, we still have some space to improve our tool, so-called tool. It's actually partly a pipeline, partly custom-developed tool, and to add GC correction for lower coverage on targets, and find more precise copy number estimation, and probably quite important is to Dockerize our tool, that it will be available and highly scalable. Well, this is our project link. Thank you for your attention. Yeah, we tried to combine like actually three ways of estimation of CNAs in order to get more reliable results. Well, I guess it's kind of higher probability that if some CNA is predicted by more instruments, it's actually there.
I understand that this would be maybe the ideal approach, but we sort of lacked time and we started to sort of dissect current approach for yield some lower resolution and just get idea what is wrong with aggregation in current available tools. Not sure if we got this idea clear, but surely in the future it would be better to combine it.
Well, it seems like most of the tools, they don't actually focus on like focal events, you know, just on one gene, and they tend to like scale up things a little bit and to predict it for bigger parts of the chromosome.
Yeah, then we move on. Our next team is the team that worked, I think the name of the team is Diablo Gene or something like this. And they worked on the HTTT assembly.
Greetings, folk. As a Diablo Gene team, we are glad to present you this project. Also, for me personally, as a non-bioinformatician, it was fascinating to work for the first time on my own source code, meaning human genome. And that's amazing that so recent findings in assembling of human genome makes it possible to analyze this data, and every one of you can actually analyze this data using a set of open source tools. Thanks these tools and the creators for that. And that it's possible for 48 hours to show you the result that we will show you. And despite that a lot of work can be still done, I'll still can pass the word to hellishly smart colleagues and devils.
Thank you. Next slide, please. Okay, so as you all probably know, the most state-of-the-art human genome assembly was for quite a long time human genome assembly version 38. And it was still not good enough, we should say, because quite recently there was a new human assembly. It was called T2T. It was called so because it is assembly telomerase to telomerase. It means the complete assembly. And please, next slide. And what is so good about this new assembly? In the previous assembly, still 8% of the genome were missing. And as Diablo Gene team, we are happy to inform you that the devil is hidden in details. And there are a lot of details hidden in this 8% of the genome. And please, the next slide. And for example, we can find a lot of new copies or some kind of copies of existing genes in these previously unresolved regions. And
we did an analysis for both the assemblies, both the old one and the new one, and my team is going to tell you how it went.
Okay, let's go to the next hell about Kamea's distribution. Kamea's is a substrings of certain length k what can be occurred in genome. And we calculated the distribution of it. And what can we see at this slide? On x-axis, the number of time that certain Kamea occurs in genome. For example, we can see peak at 219, and it means that Kameas that occurs in genome 290 times, there are 10,000 of such Kameas. And we know that these Kameas are distributed over the genome. It's not only telomere or centromere, and why it's 290 times, we don't know. And now you can see first manual GIF. You see, they divided. Thank you. So we also calculated non-unique Kameas in 2 assemblies, and it's funny that T2T assembly have a lower number of non-unique Kameas, mostly because Kamea that occurs 2 times in genome. They are only one that more frequent in Grach 38. So the next hell is.
Okay. To do this, we developed a pipeline. And what is a clinical significance of this pipeline? Okay, so in in 38 human assembly, there is some variants that have only one entity in the genome, like this one. This variant with this ID has only one entity in 38 assembly, and by our pipeline we can check how many entities in pipeline or in T2T assembly it has in how many entities it has in T2T pipeline. Yeah, like this. Like for example, this pathogenic variant has only one entity in 38 genome assembly and 15 entries. Entries in T2T. Yeah. Okay.
Okay, and I want to emphasize the importance of the analysis because there is a lot of pathogenic variants that actually exist in one copy in 38 assembly, and it has a lot of copies in T2T assembly. And you have a lot of unknown significance and likely benign, but our interest was pathogenic variants. And okay, so there were a slide with occurrences of pathogenic variants, and I have good news for you. These pathogenic variants have 2 or 3 copies in T2T genome, but actually it is really important to keep in mind that if you have some pathogenic variants in the actually old assembly, you need to these variants could be grabbed from these regions that were not assembled in 38 assembly. So these are our conclusions, and you can see the whole pipeline and scripts on the GitHub. So thank you for your attention, and yes, you can reproduce the analysis.
K-35, the first one of the series.
Okay. We'd like to check it out, but we don't have enough time for that because the alignment of that. Yeah, we checked only one peak. We checked only chimeras that occur 290 times in genome, and other one was out of scope because it really hard task for aligners. Actually, we don't know. Sure. Yes. Yes.
It's not easy now to interpret these regions because there is no good annotation for them. So it's not easy to say, you know, T2T genome, there is almost no any annotation. So it's hard to yeah. Yeah, we did this. Okay.
I think it is pretty obvious that these particular peaks that are unique for T2T, they are responsible for some repeats or tandem repeats, but actually we mapped them to the T2T genome, but we don't know, we didn't have enough time to analyze them really properly. But yeah, they are repeats, but we don't know which particular ones.
We have seen that there is, I guess, it wasn't so easy to see, but I guess that most of these chimeras from this peak are from centromere. Yeah, I think so, but it wasn't so easy. We didn't, you know, have enough time to do good statistics. Yeah.
So we split our genome to the chimeras, and then we mapped them again to the another assembly. So it was the chimeras from one assembly, and we mapped them to the another. And actually, this particular chimera mapped to the 15 different positions in T2T. So it is pretty easy, and yes, we have. So this variant could be mismatched. So you can not recognize this particular variant because the reads could came from another regions if you use kit with 75 base pair long reads. So this analysis was performed for 75 long reads. No. it is this variant, yes.
Can you say it again? Pathogenic, it's from CleanVar database, and it's usually associated with some diseases. You can open it and read which diseases, and this one associated with one rare blood disease. Yeah.
Thank you. Have to move on. Yeah, and now switch to the team that worked on the issue of protein stability, I think. Yeah, so please come here, present. Yeah.
Thank you.
So hello everyone, and our team is happy to present you our take on protein stability and whether pre-trained embeddings are good representations for that task. Sorry. So this is our group leaders online, and that's us. Well-trained llama Barbies, and we want to explore protein stability as one of the most crucial features of proteins, and how can we assess that feature? We can assess that feature, for example, with the help of flow cytometry and labeled proteins. And in this kind of experiments, stable proteins tend to be more rigid under their trypsin or other protease exposure, and so we thought that it would be more efficient and definitely cheaper to retrieve that information from the protein sequence itself. So our plan was just pick one of the large language models, explore their embedded space, solve protein stability as a metric learning task, and compare it, of course, with other researchers, and finally probably understand what and why we did, but here it is.
Yeah, during this task we face a lot of problems, the often one was the out-of-memory CUDA error, because we firstly work on the server, not on the cloud. And there are a lot of other errors that we faced, but we figure out what it is, and we want to thank Prostata because we retrieve a lot of code insights from their codes. So thank them. So what about the data? We work with the data from Rocklin et al. 2017. This data set, there are two data sets, one from single mutations. So we have wild type sequences with length 43 and mutated sequences. So in first data set we have only one mutation. And in the second data set we have multiple mutations, so from 1 to 43. Yes, and we have our label, it is a stability, the delta stability, so we measure the wild type stability protein and mutated protein and retrieve the differences. So there are the distribution of delta stability scores. So how it, several words about stability scores. So it's according to the Rocklin paper, it is a difference between the measured 50 efficient concentration, measured efficient concentration and predicted efficient concentration on the specific models. But not all the data are ideal, and so in the multiple data set we see this strange picture. So we have a lot of data with one mutations and several, mutations a little bit, and a lot of mutations that, so I remember you that it is the length of the sequence is 43, so it is like almost all sequences are mutated, and it's, so the stability score in this, the delta stability score makes no sense in this, so we discard this data. So next. Yeah.
Having two sequences, we use transformer-based model for extracting protein features. It's ESM2. We designed two models. One of the first is used embedding only for mutated positions, and the second one is embedding on the whole sequence. After that, we concatenate these embeddings to input to linear layers to predict stability score. Yeah, we benchmark our models with existing solutions. Models that used embeddings only for mutation positions show better performance. In order to increase model performance, we add additional data having multiple mutations, and it shows worse results. We drew a correlation plot of our best model.
Okay, and then we decide to draw t-SNE plot to see maybe more correlation, but not. And how we do this? We retrieve embeddings using pre-trained models, and after that we draw this plot. But it is okay that we can see some correlation in this plot, because we can see correlation not in 2D dimension, maybe in other one, because embeddings have more significant dimension. And finally, we fine-tuned the model, and this model showed a good performance, as we can see in benchmarking table. Then we understand that extracting embeddings for mutated and wild-type amino acids outperform if we extracting embeddings on all sequence. And finally, multiple mutation dataset don't improve the model and contain some contradictory samples. And finally, follow the QR to retrieve our code and data, and follow llamas on the lam. And some questions, maybe. Yeah. Yes.
Yes. We used embedding or tokens, no, amino acid only in mutated positions and in corresponding positions in the wild type. Yeah.
Yeah, but like, it sounded like you kind of used two of them at the same time, right? So like you took mutated wild type, put it together into a linear model.
Yeah.
Have you used just mutated as a predict?
No, we can concatenate them separately. We're not used. Good point. Yeah, we haven't done baseline. Yes.
Okay, thank you. I think we should move on because unfortunately we are already after that which we have ended this session. Yeah, thank you.
Yeah, and next we have the team that worked on subtyping of small cell lung cancer. I think so. Yeah.
let me have a go. Yep, it's working. Hello everybody, my name is Anna. Me and my team are presenting our project based on the clustering of small cell lung cancer, in short SCLC, based on 2 different data sets, data types, including the expression part and the microenvironment. To start with, we extracted the data from the 2 papers published by Julia George in 2015-2018. First of all, this data was that was the 2 different data sets, and we had to convert it to one format, which is the TPM. And after this, the data must have been transformed, normalized, or standardized, and merged together. Taking into account that the data sets were from the separate papers, we did observe the batch effect, and to correct the batch effect, we
yeah, considering the fact that there was 2 different papers, we expected our samples to be heavily batched. That's why we did the scaling, and after scaling we also did the batch correction with using Scanorama. However, it was not that much of a difference, so we proceeded with the just normal scaling correction.
Yes, after all, we had a huge merged data set with around 20,000 genes presented in the expression matrix. That was quite a large set of genes, and we decided to filter those genes on the to get the highly variable genes that could have some amount of biological significance. And for this purpose, we thresholded all the genes based on the standard deviation of the specific gene expressions, and we yielded around 10% of all the genes, as you can see from the right side of the threshold. And according to this data, according to the clinical data, out of those highly variable genes, there were also genes with the high proved clinical significance, around 150 genes that were associated with lung cancer. So here we present a set of significant genes, and we were working mainly with highly variable and clinically significant genes in our data sets. And starting with this data, what we did first is we clustered them hierarchically using the Euclidean distance and linkage complete on the completely batch corrected set. And as you can see, the hierarchy shows us that the sample split into 2 divergent groups, 2 divergent clusters, at least 2 divergent clusters. But we also can see the splitting, not at the root, but several nodes. So we started wondering, actually, how many subsets and subtypes of the lung cancer cells there could be based on this expression data. So for this purpose, we proceeded with the PCA analysis. We constructed the 3-dimensional PCA based on the data, and we saw 2 evident clusters. As you can see at this cluster, we have samples that have the high expression of the PO2F3 class and non-PO2F3 class, and they do split into 2 divergent groups, not 3, not more. And we also developed a metric to see whether 2 subtypes are enough and as an optimal number of the subtypes. For this metric, we used the silhouette score of the subtypes in the PCA 3-dimensional matter. And as you can see, yes, we yielded 2 subtypes mainly. And starting and having this in the account in our mind, we were wondering, does it have any biological significance if the data splits into 2 main clusters? So with this in mind, we decided to see how those 2 clusters differ in terms of the expression. And in the first cluster, there are upregulated transcription factors that are associated, and also the transcription factor of ASCL1. And in the second group, this transcription factor is downregulated, and PO2F3 is upregulated. So we can see that there is a huge difference in terms of the expression in the genes.
We then wondered if the same
division would be seen in the convolution data. So we ran through the APIC software, and we saw no division in two clusters. We saw only two outliers that were purely of immune cells and had no tumor basis. And therefore, we continued with some other type of biological classification and moved on to...
Yeah, we looked at the differential gene expression, which is not the name of the slide, but we had, using Mann-Whitney test, we selected for top 20 genes that are the most significant, significant difference. And as we expected, most of them are just transcriptional factors. However, it's interesting to see that they have completely different roles from a biological point of view, even non-related to lungs or anything else.
So we so probably the main result of our work is that we trained a classifier. We used predictions of a clustering model as labels, and then we predicted on an independent data set, and you can see that the classes, yellow one and green one, correspond with the two distinct areas on a heat map, so we conclude that our classifier is precise enough.
So overall, we satisfied our aims mainly. We did see the two clusters. These clusters, they differ in the neural differentiation genes, the transcription factors, and they don't differ at all in the deconvolution. And based on this data, we managed to train the classifier. And yes, there are areas for further research, and we are really grateful for this setup and to have the opportunity to participate in it. And the note for charges, we really enjoyed this. We are the youngest ones here. And also to learn more about the code and the project, go on a GitHub. Thank you. So the aim of the task was actually the lung cancer is incredibly hard to tailor the clinical approach to, and therefore we just noticed that from the background reading we saw that there are several subtypes that have to be treated differently, and based on the transcription factor that is affecting the lung cancer development, we can tailor the treatment approach. And also, this exact gene is really relevant for us because, as we saw, the papers are not published, but it is mentioned in the in the preprints. It is mentioned in the preprints that actually first subtypes diverge based on this PO to F3 transcription factor, and then they develop into several other subtypes. And this might be a key into actually discovering what the best treatment would be. Yes, this one is described. It has the relevance to development of the lung cancer. Not positive and negative, but different clusters. Yes, yep, yeah. Mm-hmm.
Okay, so like, how many of the cells are positive in terms of like percent? Like, you took several samples, and then you show like on a PC, for example, you have like very few that are positive, and like very many that are negative.
Yep.
And this looks to me like a very unbalanced data set in terms of like how many samples present that are positive.
In this case, we did not focus very much on this because we initially wanted to go further and to subtype it more. But answering your question, from papers, it's seen that mostly it's quite balanced. So in terms of tumor itself, the cells in numbers are divided into more or less equal subtypes. It's seen on the scans, it's seen on the tissue samples. But yes, as you pointed out, here we have a bit of unbalanced cell number clusters in terms of...
No, we saw the divergence in the big group subtypes, in the big subtype. We didn't delve deeper into this because we knew that there is a big significance into 2 peaks, and maybe for the further research we're going to remove the big peak with the positive cells and delve deeper. But at the initial point, we do see the divergence into 2 groups, which we wanted to investigate. Thank you.
Thank you. I'm sorry. Let's move on. Yeah, so next team was working on a project related to coronavirus evolution. Please.
Okay. Biostatistics, dear audience, I hope you are here with us still. My name is Tatiana and here's Daria, and we are ready to present you our project about epistasis in really huge dataset. So, a bit of intro. I think you all remember what epistasis from school, yeah? Interaction to proteins and result a different color of the food. Actually, no. In the evolution, there is much more wide definition of epistasis. This is about interactions of 2 different alleles or SNPs. As a result, the relative fitness grows up if they are together. And actually, we tried to check several hypotheses we generated through this study. And first, we wanted to check where epistasis most likely to appear inside gene well close to each other, or for example between different genes which products interact with each other. And also we were interested in indels and SNP difference in this process. Well, actually this problem can be named solved because there is different solutions how we can calculate it. For example, the same models, and there is well, small link to a really good article with great math, but the work were done on proteome, and we desire to study DNA SNPs. That's why we wanted to, well, set up something from the 0 point without any tools. We took our genome, called SNPs, and first thing we obtained such blanket. What you can observe here? Well, there is 2 coordinates, each of them is COVID genome, 30,000 of nucleotides, and intersections is the well, presence in the same certain genome. So the more genome with 2 these mutations, the brighter dot will be. So here it's not really clear, but we bent it a little bit, and we can see, for example, that the most bright part on the end of genome, obviously spike protein and other structure proteins located here. But also we can observe some interesting dots. For example, this in the middle between NSP3 and NSP12, and their products actually connected structurally, but well, it's difficult to see how certainly. And for example, there is small triangle inside NSP12, and we assume that we showed some epistasis inside one gene. Well, looks similar, yeah? Like Hi-C map. So we decided, why not? Why can't we try to, well, convert our coordinates to Hi-C map format and try to implement some Hi-C tools? For example, for TAD inference, well, in different meaning, like the here. In different meaning here, like searching interactions, but with tools already existent. And at this point, we collabed with another team. You will listen the presentation a bit later, and they're innovating new tool for TAD searching, and we just tried to implement it. Well, for example, there is definitely seen TAD, but not really good observation of small TADs, as we expected to see, but probably later we will try to change some parameters and get the picture we want to see.
Yep. Can you please scroll the slides, because, yeah.
Johan did it. Right, so we found this method of displaying the data, and we thought, well, we have time data, and can you please turn on the GIFs, someone? Yeah. Yeah, so what we did, we made a timeline of appearance of SNPs through the genome. And what you can see 2 years, 220, 2020, and 2021. So what you can see, we can see straight lines appearing, and we can assume that these lines appeared, they started appearing in summer, and can see it appearing in autumn 20, and we assume it's Omicron strain, which was fixed in the population. Next slide, please. Also we checked this hypothesis, whether these lines are indels because they may lead to frame shift. Apparently not. We deleted all indels from the data, and the lines that did not disappear, ergo they are not indels. Next slide, please. And after that, we tried to remove all the most abundant SNPs to find more, hidden patterns. And it appears that the upper part, how it's called?
Structure proteins and spike proteins.
Yeah, the region with spike proteins has a lot of SNPs. It looks like it is connected to the mouse genome, and it's likely that, well, I can't say likely, but yeah, we see that a lot of SNPs happens there. Back to the presentation, please. Yeah, last slide. Sorry. We tried to do some phylogenetics, but let's skip it. Yeah. So we tried to find the epistasis. We found a new method, and we think it is a great start for discovering new patterns in epistasis, and we hope to work on project more. We have our GitHub link on the next slide. Yeah, but yeah. Yeah, thank you very much for your attention. It predict, I mean. Yeah, we hope to do that, but we need more time for that. Multiple ways. We can use some machine learning methods, for example, because you see that's images. We can stack them. We can apply some learning models that use images as an input.
Actually, we have also quite big metadata for this data, so we can pre-train on this and probably try to predict something more. Yeah.
Another way is like, I don't know, classification, for example. We try to find some features. We can try to, I don't know, a lot of stuff can happen. Yeah, we just need more time.
Thank you. Let's move on. Next team was working on the aging clock, I think. Yeah, please. The floor is yours for your 5 minutes. We'll be limiting the discussion to one question, I think, because, again, we have very short on time.
Thank you. 2 days ago, we was more maybe crazy or enthusiastic, and we think two types omics, it's not multi. 1 second. We need a tree. Because metallome and metabolomet is good, but hole between needed transcriptome. And we have very big data sets, more 10,000 samples. And what we want to find, we want to study specific points in the age where aging a lot of changing. in the theory, we thought about 37 and 60. But we need to check it and try find this points and maybe time in our data. And the first we use windows and cut metal on data in the window where we calculate correlation and then see how this correlation can correlate or maybe between these points. And yes, there is two maybe windows in these types who a lot of change in CPG sites. Then big data, it is big trouble. We wanna filtering data and find CPGs who are more changing between the first and the second group and other groups in the aging. In mutilation can change a lot of covariates like cellular components, calendar age, and experiment like batch and gender and other, and we make construction where we try add a lot of there, and we find a lot of CPG sites who was between the first and the second group, and less in the third, and 0 in the third and the second and the third. Yep. These CPG sites help us separate the first group in the other, and then we start work with expression data and make special algorithms where we try find genes where changing angle in these three groups. It's model including batch effect and other covariates too, and then we integration transcriptome and metabolome and find genes and CPG sites who including in this two group, but we yes, and we see the similar changing in the gene and CPG sites who was around there. And then we work with metabolome data processing, and then we use reactome, reaction database, and find what the reaction including this gene and metabolome, metabolic, and CPG sites, and what? Yeah. Yes, that's all. And then we find 7 it's not process, it's reaction, yes, it's reaction, who work with cholesterol metabolism, energy production, and cellular signaling, and this mechanisms, including in the aging before. It is more about this. 1 second. Yep. And that's all. Thank you. You can use GitHub repository where we add information how we can use these methods and data. Methylation part, okay.
Like you adjusted for the cell types, for the groups, for the...
Yep.
I think for the sex of the donors as well.
General, yep.
There is
This?
no differentially methylated CpGs 2 versus 3 in comparisons. Like, why do you still believe that there is like 3 phases? You show the results right there. So like, show the, like in comparison 2 versus 3.
No.
Previous slide.
Okay. What?
In comparison 2 versus 3, there is 0 CpG differentiated.
Yep. And
Differentially methylated CpG ions.
Yep.
So like, why do you still believe that there is like 3-phase model for aging?
Because these 3 groups and in these ages changing in other omics. It is information in the literature, and we know about there is three groups in this times, and in the line between changing angle or changing process can, but we wanna find this change in the methylation and expression. In the expression, it is more, you can look it maybe more effectively, because, but methylation usually changing like random and changing with a calendar age, maybe more correlation with these parameters, but didn't have a lot of strong these changes. But we including this 3 type and can find this maybe not a lot, but very improved points where there is this changing.
So one more question. Like,
okay.
There is like huge difference 1 versus 2. There's like almost no difference 2 versus 3. Why there is almost no difference in 1 versus 3?
If we talk about how it looks in the other parameters, usually in this.
No, just like
no, yes.
This looks like a typo to me.
What?
This looks like a typo. Like you report a lot of differences in 1 versus 2.
Yeah.
Almost no differences in 2 versus 3.
Yep.
And then you compare 1 versus 3, and you have like order of magnitude less results.
Because how to say, usually 0.3 in this, it is this balance, have more diversity in these points, and you not all of them can find maybe good correlation, or maybe.
Sorry. Yeah, we have 2 teams left, so please stay strong. Yeah, the next team is the team that worked on High-C Copilot.
Okay. Just a sec. The usual problem. Sorry. Okay. So, hello everyone. I'll present results of our team, which called Heat Shock Jara. because we are all enjoyed our time in Yerevan looking at heat maps. So our project name is HiC Copilot, and the purpose of the project is to create a tool to improve HiC guided genome assemblies. HiC data is a kind of data which shows interactions between different parts of genomes, and it has multiple applications. One of them is genome assembly. So the genome assembly can be improved by using HiC data by several ways. For example, it helps to locate and orientate contigs, to detect borders of chromosomes and centromeres. Also, it can help to identify different misassemblies or structural variations, such as translocations and inversions. And sorry, it's okay. I'll be more tender. Yeah, and the last thing, often in real life requires a manual curation, which basically means that the person sits and look at heat maps a lot. So I'll try. I hope to show you how it looks like, but it's sorry. Okay, so for example, this is like a normal heat map showing the interactions between different parts of genomes, and the red dots basically shows the places where interactions occurs. And yeah, thanks. And doesn't like me for a reason. So I'll delegate it. Can you go back, please? Yeah, this is back. Yes, so this is how inversions look like. This is normal, like normal heat map, and if you can make this is an inversion, and we need to dedicate a lot of time and a lot of training for a person to be able to detect it. So we created a tool. Next slide, please. To detect different misassemblies. Just stop. And we used several reference assemblies, high-quality assemblies with different genome size, just to create like controlled data sets with errors. Next slide, please. And we simulated inversions and translocations on this heat map HiC data with different size, and they were located in random places in genomes. And we used this data to test our tool. So now we know how to break the genome, and we need to know how to fix it. But yeah, you can see that. But to fix, before we starting to fix anything, we need to locate the error. And what exactly can we detect? Can you? First, we can find borders of high zones of high interactions. And how we did that? We went with sliding windows through the matrix. And we were looking for local minimas, and local minimas located exactly on the borders of the squares on the diagonal of the matrix. So so our tool is able to locate the squares, which correspond to chromosomes in this case, but it also can be topology associated domains and other regions of interactions in genome.
And then we tried to detect translocations as well on our simulated data. And how we did that? We went with sliding window not through the diagonal, but through the entire matrix, and we look for the points where the counts rapidly increased. And now we are able to detect translocations, which are looked exactly like this. And we didn't start to work with inversions, but we are on our way. And also we tested our tool on the COVID group project, so thank them for trust. And this is it. Thank you. Yep.
Thank you. You moved with a sliding window, but with such an approach, the size of the window really matters.
Yes, that's yeah. Sorry.
Yeah, do you test it? What was the results?
Yeah, we tested different sliding windows, and we just empirically selected the one. We selected size of 150 beans in this case, but it really depends on the data set, and we didn't come out with the optimal, like with, yeah.
And clicker. Okay. Yeah, the last team, last but not the least, as usual, the team that worked on a single cell RNA sample representation. Yeah.
Hi everyone, my name is Amina. The ladies are Sveta and Lila, and we are going to present the results we achieved on this hackathon. So the main aim of our project was to apply the contrastive learning approach for representation of single-cell RNA sequencing samples. During the last 48 hours, we created a method for statistical interpretation of obtained representations. We performed these representations using a baseline method and a contrastive learning approach and compared them. Well, so in order to do so, we several tasks were set. You can see them on this slide. And we also used, the next slide please, we used annotated COVID-19 single-cell RNA sequencing data from 140 donors. These data were obtained from COMBAT consortium, where COMBAT stands for COVID-19 Multi-Omic Blood Atlas. And though in this consortium a lot of different data types were obtained, in our project, in our research, we focused specifically on single-cell RNA sequencing data because they allow researchers to describe cell variability with a very high resolution. The next one, thank you. So, in order to do so, first of all, we developed a neural network model to learn distances between samples using a contrastive learning framework. Contrastive learning uses the principle of contrasting samples against each other to learn the attributes that are common between some data classes and the attributes that set apart one data class from another. For each of 140 donors, we generated a pair. We generated a pair of data sets of n cells, where n is a hyperparameter. Every such subset was collapsed into a single row. So basically, we turned single-cell RNA sequencing data into pseudo-bulk data. This pseudo-bulk data is an input for a neural network which compares positive and negative pairs. The network learns on these data sets. After that, we use comprehensive data sets that contain all these cells for each donor, and the output is a matrix of distances between donors. After that, we tested developed algorithm architectures with different depth and width and hyperparameters, such as the number of cells per sample and batch size. You can see some of them on this slide, the hyperparameters. And finally, and finally, we compared distances from a learned representation with distances calculated using baseline method, which didn't involve learning. For that, we used Leiden algorithm for clustering of samples and applied statistical testing using chi-squared test to find out if the biological variability, for example, outcome for the patient, and the technical covariate like institute where the samples were collected and prepared differed between obtained clusters. And we'd like to note that the representation is obtained in purely unsupervised way, meaning that the model had no idea about outcome or other patients' covariates. So we came to such conclusions. On this slide, you can see comparison between baseline method and the best and the worst of learned methods. The best and the worst of learned methods were chosen by their ability to see connection between the outcome for patient and between clusters. As you can see, baseline method was able to see such difference, while even the best of learned methods couldn't. But the best method captures biological variability, though it is affected by technical covariates like institute. So basically, the result is that baseline method works very well, but more work is needed to beat it using contrastive learning approach. And we could also highlight that the models with from 3 to 5 neurons, batch size of 64 and 1,000 cells of sample representation work the best. The parameters of the best and the worst of learned models are on this slide. That's it. That's you can, our code is now stored at GitHub and the link is attached. And finally, I think that the main result our team achieved at this hackathon is that we met a lot of our friends and had the greatest time, and I'd like to use the opportunity to thank all the organizers and all the participants of this hackathon, and we'd also like to say thank you for our great project leader Vova Shitov for his endless love and support and help. Thank you very much. Yeah.
So, as far as I understand, the order of cells matters when you choose these blocks and make comparisons. Like in the picture, the order of rows matters.
Maybe my co-workers will correct me, but as far as I understood, no, it didn't. The subset of cells was important. And we changed the number of cells we would subset from it, but the order would.
Was there any grouping of the rows?
as far as I know, no, there wasn't, because there are just different cells for each donor. So, no, yeah, yeah. If I understood the question correctly, the size of pseudobulk data is a single row. So the main idea is to transform the matrix into a single row for each donor, for us to be able to learn the model and yeah. No, the number of cells we used, it was a hyperparameter, so in different tests there were different numbers of cells we would subset from the data set. So yes, sometimes it was 100, I don't remember. Ты помнишь, на каком слайде типа гиперпараметр? Yeah. Yeah. Well, we make this subset of some amount of cells, like for example 100, but we used different numbers of cells too, and we collapse them into one single row. As far as I remember, we used я забыла слово среднее. Yeah, the mean statistic. So, and we need this procedure at the point where we learn our model. So we take these subsets, and since we at this point know from which donor these subsets come, we can say to our model that see, these subsets are from one donor, so they should be similar, and these from different, so they should be different. And later, yeah. Yeah. And we make this procedure a lot of times with different number of cells. We make these subsets, as far as I remember, they sometimes can even be repeated, some of these cells. And we make this procedure, we learn our method, and then when we have this architecture, we use all the cells. Yeah. Thank you.
If you can make it briefly, yeah.
What's the baseline method?
Может быть, кто-то мне поможет объяснить? Okay, sorry. So the baseline method was, it was basically the same idea, but we didn't take these subsets. We just used all the cells. We took the mean values for them, so we turned single cell RNA sequencing data into a pseudobulk, and we didn't put it into a model. So we just counted the distance matrix, and that's it. And we compared this baseline method with that approach that I was explaining, where we used the model, and the model helped us to make a предсказание. I'm sorry. Yeah, about, yeah. Yeah. Thank you very much.
Yeah, and with this we're done. Let's make a round of applause for all of the teams who presented their results. Yeah, thank you. And now the jury will need around half an hour to discuss basically the results and reach a conclusion regarding the prizes. So I think we can meet around 8 p.m. So let's set 8 p.m. as a time point, and then well, maybe it will be like 2 or 3 minutes later than that. But
Ну давай тогда ты, получается, там же 5 биоинформатических, 1 биостатистический проект.
Ну, 2 5.
А кто?
Не надо. Вначале не надо этого точно говорить.
Нет, я не собираюсь об этом говорить. Я просто, а какой второй биостатистический? Да. Да. А нет, подожди, мы же, у нас 7 что ли теперь?
Все.
Я просто...
Раз. Да, чего? Одну маленькую секундочку.
Я начну, потому что 7-й статистики последний.
Давай я скажу как бы это, что... Ну или можно. Давай.
А вы на каком языке?
На английском, я думаю. Okay. Yeah, good evening, everyone. We are ready to announce the prize winners for the 2023 Bioinformatics and Biostatistics Hackathon. It was a tough choice, but I'd like to say that this time the jury was well, very consistent in their assessment. And first we will announce the 4 special prizes, which are not the top 3 of our choice, of the jury's choice, but are still, I think have done a very, very nice job, and we'd like to award some special prizes to these teams. Yeah, and having said that, I'd like to give floor to Eugene to announce the first prize winner.
Okay, and the first special prize goes to one of the... Yes, so we have one big special prize, but the first part of this big special prize, anyway, goes to, and for me it's a great honor to announce this team, it's Hammer Hazard. Please.
Okay, all 4, yeah. Yeah, so... also in this special prize group, we have the Wings team. Yeah. Next up is the team which didn't have a special name, so it in our own assessment it goes by the nickname Wova's team, which basically means that these are the guys who did the contrastive learning project with Vladimir Shidov. Yeah, unlike the project presentations when you had to basically just defend yourself, now you can ask and invite your project supervisor to the floor.
But he has to keep silence as well.
Yeah, and finally, in our special prize winners, we have the Hidden Dance team that worked on coronavirus evolution project. Yeah, so these are the teams that won the special prize. Yeah, thank you. I think you can take your seats. Thank you so much. Yeah. Yeah. And then I'll check once again, not to be mistaken, you know. Yeah, so then we move to the top three, and we'll announce them from the third place going to the first place. And the third place of the Bioinformatics and Biostatistics Hackathon today goes to another team which didn't create their own name. So I'll have to just tell the topic of their project. So this team worked on the subtypes of small cell lung cancer. Yep. So now we move to the second place. Yeah, I have to say that the fight between the second and the first place between these two teams was very, very tough. And the jury had a hard time deciding who gets the first prize and who gets the second prize. But unfortunately, there was only one winner. We'll start with the second place. So the runner-up for this year's biohack is the WOOF team that worked on the O antigens in bacteria.
And looking on my smiling face, I think you can guess to which branch of science does the team belong who get the first place. And the first place goes to the team Tryptophan.
I think we are ready to stop the translation. Mush?